So welcome back everybody and welcome to the new faces. I think we are now heading towards a session we are very much interested in, and that is using devices which are drug eluting, and that is something we're looking very much forward because there are still some non-resolved problems, particularly in the frontal, where we are dealing not only with inflammation of the mucosa but also inflammation of the bone or of the periosteum, which we cannot deal with surgically or very difficultly. We need to address that from a medical point of view, and I think drug-eluting stents or devices might be helpful. I'd like to invite Susan for her presentation on the device.
Thank you. I'm here today to speak about the Propel steroid-releasing implant. First of all, a little bit about Intersect ENT. We are a company that is dedicated to developing local drug-releasing implants. Our first implants are dedicated to treatment for chronic sinusitis patients. What really started Intersect ENT was the belief that there was a better way to deliver steroids directly to the tissue. The core engineering team from Intersect ENT had developed drug-eluting stent capability and technology in the cardiovascular world. Myself included, I have a biomedical engineering background. And we thought, when we saw all the ways that patients were, completing irrigations and sprays and nebulizers and taking oral medications, we knew that we could bring this technology to ENT surgeons and their patients. The premise of the device is really based on some of Dr. Kennedy's research that shows that really optimizing wound healing postoperatively and preventing scarring and inflammation postoperatively can set that patient up for the best possible long-term result. And so our device is really designed to prevent such scarring, middle turbinate lateralization, adhesions, and inflammation and polyps in that postoperative period, again, in order to set that patient up for long-term success. Propel is a bioabsorbable steroid-releasing implant. It is self-expanding. It is the first and only product backed by level 1A clinical evidence. And in fact, it's the only combination product for use in chronic rhinosinusitis, and over 125,000 patients have been treated to date. Again, our goal is really to maintain surgical outcomes, so to protect the sinuses that you're treating surgically from recurrent inflammation and recurrent scarring. And in doing that, really help protect and maintain the work that you're all doing in the operating room. The way that the product works is a sheath is advanced into the cavity. The Propel, again, is self-expanding, and so it springs open. It naturally conforms to the individual and unique shape of that patient's sinus anatomy after surgery. And the steroid is delivered over the course of 30 days. The cavity is then maintained after surgery, and there's a short video that shows the deployment of the device. And as you can see here, again, post-ethmoidectomy, the surgeon advances the delivery system into the cavity. The Propel is deployed. It supports that middle turbinate and also then delivers drug throughout that entire ethmoid cavity, again, over the course of 30 days before dissolving over the course of 45 days. Why mometasone? Well, really, it's the optimal drug of choice, and I think Dorna will agree with that. When we looked at this early on as an engineering team, we were looking for a drug that's lipophilic, so it readily absorbs into tissue and not water or mucus, for that matter. That it's got a very high glucocorticoid receptor binding affinity, and in fact, mometasone furoate has the highest glucocorticoid receptor binding affinity, making it very potent. And then, of course, it's safe. And what that results is in choosing that drug and delivering over the course of 30 days is that drug actually remains in the tissue at therapeutic levels through 60 days. This is just a slide showing the comparison of mometasone compared to some of the other steroids that are readily available in liquid form to ENTs. And when we looked at these, you can see dexamethasone, triamcinolone, budesonide that sometimes are available for treatment of patients either via irrigations or sometimes mixing with packing materials, were an older generation of corticosteroids. And important to us was to choose one that was of the more advanced version to maximize the potency and the ability of the drug to get into the tissue itself. We are a company that is dedicated to evidence-based innovation. This is the only device in sinus surgery that is backed by level 1A evidence, not just combination products. And in fact, it's the only medical device in ENT backed by level 1A clinical evidence. There are 4 prospective multi-center trials totaling 285 patients. 3 of those are randomized, blinded, controlled, and grading was completed by an independent blinded reviewer. So not only was the patient blinded, the treating clinician in the ethmoid studies were blinded, but then the video endoscopies were also collected and then reviewed by independent panel of blinded reviewers as well. Why is that? Well, these studies were designed not post-operatively, or sorry, post-market, but they were designed to support FDA approval as a requirement for such a Class 3 medical device. And what we've shown is statistically significant reductions in the need for post-operative interventions, both in the ethmoid sinus and then similarly with the frontal sinus treatment. The ARS recently has issued a position statement in supporting of Propel and mometasone furoate implants in general, and has stated that they feel strongly that the product should be available to patients at the discretion of the physician, of course, in order to maximize outcomes. Beyond that, our commitment really is to continuing to develop various devices for local drug delivery within ENT. We look forward to collaborating with all of you and continuing to collaborate on furthering this technology so that the patients that you treat, as much as possible can, reduce their need for taking systemic medications. Thank you.
Thank you very much, Susan.
Today I want to present BioInspire Technologies to you guys. You might not have heard about us, so I would go a little bit more into details of what our technology is and what we hope to achieve with our technology. So the technology is really a film that can stick to any tissue or organ. So specifically it can stick to mucosa. The film is very thin. As you place it on mucosa, it absorbs moisture and it adheres to mucosa and remains in place. It can act as a physical barrier, for example, to prevent formation of adhesions. It can also incorporate various therapeutics or drugs to deliver them locally before being completely bioabsorbed over a period of time. I want to show you a little bit more about the work that we have done. And this is how the film, we call it Sinband, works in the sinus. As you can see on the left-hand picture, that's the yellow is the film. It completely conforms to the cavity, the roughness of the cavity. On the right-hand side, you see that in an actual patient. Again, we pretty much wallpaper the entire sinus. The interesting thing about it, because it conforms, there is no protrusion into the sinus. Also, the patients can breathe easily. There is no packing material. From patients' perspective, they never know there is any device inside their sinus. It's completely convenient to them. So we have done significant amount of preclinical work in terms of proving the technology, how biocompatible the technology is, how it does not induce inflammatory reaction. We've looked at, I think, more than 100 rabbit sinuses where we implanted the device in. We look at tissue response by looking at histology. We also look at drug release kinetic from the device. I'm going to fly through the next few slides very quickly. This is a histology of the device. Sinband implanted 48 hours in rabbit sinus. It adheres really well to the wound. It remains in place. By 5 days, you start seeing epithelium growing underneath and on top of the film. By about 9 days, the film is completely epithelialized. So there is, you can see new epithelium grown over the film. Really what we think happens is the film becomes like a submucosal depot and remains under the mucosa, continuing to do what it's supposed to do. By 15 days, there is no sign of the device. It's completely resorbed. Throughout all of these studies, we never saw any foreign body reaction or inflammation caused by the film. The film is made of fibrinogen, so that's expected. This is a natural material that does not cause any foreign body reaction. So this is a drug release into the sinus. The drug incorporated in Sinband is fluticasone propionate. I do agree with you. These are the second generation steroids are definitely the way to go in terms of local delivery, as they're designed for local delivery. What we have done here, the films were implanted in the sinus. We've collected the mucosa, and we've looked at the concentration of the drug in the mucosa. We also looked at plasma levels, although they're completely different levels, but you can see they follow the same trend as what you see in the sinus mucosa. By day 30 and 44, there is still therapeutic levels of the drug within the sinus, although by day 15, we never saw the film again. So that kind of goes back to the theory of the film becoming a submucosal depot. One of the questions I think was raised to this panel was what happens at higher doses and what do you get? And it's an interesting question because if you increase the dose, the question is more drug going to come out immediately or are you going to have a longer term release? So what's the impact of increasing the drug? So we set out to figure that out. We created Sinubands with various doses of steroids. We implanted it again in rabbit. We looked at them at day 7 and 19 where we collected the tissue. We looked at it histologically. We also looked at plasma levels. Again, these plasma levels are extremely low. We actually had to go out of our way to find very precise measurements so we can even measure them. And remember, these are really small rabbits. So in humans, you would never see anything show up in plasma. So what we saw here was at 7 days, so these are very different doses, increased doses. At 7 days, they're pretty much very similar levels of drug released. But if you look at 18 days, you see a very clear dose response. As you increase the dose, there's higher amounts released into plasma. Basically, what it shows is when you increase the dose, not that you're going to have more drug immediately, you're also going to have a longer-term release of the drug. We also looked at it from a safety perspective. This study has never been done, although physicians tend to use higher doses of steroids, but the effect of it locally on local mucosa was not evaluated. So we looked at all of these samples histologically. We never saw any toxic effect from the drug, no matter how high we went up, which is kind of consistent with probably your experiences. And there was no safety or inflammatory reaction because of the film, even when it was implanted without any fluticasone. We have already completed a clinical trial with Sinband. It was a controlled randomized double-blind study. The data will be published soon. I don't want to disclose the outcome of it as we wait publication. We are planning to do larger clinical studies. Most importantly, I want to present this as a tool, as a new way of experimenting, not just with the steroid, but other therapies that you guys might be interested in, other local therapies that could really maybe lower the cost of some of the other therapies that you're looking at. So kind of this is the first step in a pretty interesting, adventurous process of coming up with newer technologies and ideas to solve some of the most difficult problems. Thank you so much, and I bring it back to you.
Well, it was very good for both presentations. I really enjoyed. I mean, in previous sessions we have been had wishful thinking that was no more surgery but only medical treatment. But we are not that far yet, so we still need to do surgery, and we will certainly need some of these devices, which look pretty much. What would be your expectations in using drug-eluting devices, Claire?
So I work in a very financially constrained environment in the NHS in the UK. And I'm not allowed to see my patients for 3 months after sinus surgery, at which point I see relatively low rates of adhesions, and obviously we have no need for debridement. So some of the big drivers for using these devices post-operatively is to reduce debridements and to reduce synechia. I don't think that is a huge problem in my practice, or at least not adhesions that adversely affect the patient outcome. I think the real place it would have a role for me would be in the frontal sinus, where topical drug delivery with irrigation is actually very difficult to achieve. So I think the ethmoids and the maxillary sinus we can get to with irrigations. I think the frontal sinus is very difficult, so I think that would be an area that I would like to look at in more detail.
Mm-hmm. Peter, would like to add something?
Well, from my perspective, I think what we heard now is very promising, and it may reduce the need for oral steroids in the post-operative care of patients with or patients that have undergone sinus surgery, because in our institute it's still common practice to provide these patients with a prescription of 2 weeks of oral steroids after sinus surgery. So I think these drug-diluting stents may be a good alternative, reducing the risk of adverse event. What I would like to get a better insight in is the studies that you have performed, the duration of follow-up of the patient, because actually what matters to us is the long-term result and the long-term effects that you may achieve with your drug-eluting stents and devices. So I also see a potential benefit in those patients with chronic rhinosinusitis, where you would apply these stents as a kind of alternative for the spray and douching. So these are my 3 major comments and questions also to you, but I would like to hear your feedback later.
Would you like to reply immediately to the post-op douching and rinsing? Are there any concerns by having positioned one of your devices with a douching and rinsing with saline solution?
We've not. We actually encourage saline rinsing post-operatively in line with the surgeon's typical regimen, and have not seen any challenges with that combination. And from a management perspective, I know you mentioned the possibility to place in patients who perhaps have recurrence. We've now completed one study and published of 100 patients, and now a second study that's just received top-line results of 300 patients, randomizing a Propel-like device placed in the office for recurrent polyp patients versus a sham control, and have seen statistically significant reductions in the need for or for symptom scores, as well as reducing the number of patients that are indicated for revision surgery with those patients. And so we are very excited about that. I think Propel, this is our first step. And I'm encouraged, to see, I think we're partnering together because we believe in the future of localized drug delivery. It's up to each surgeon of how it fits in your practice. So Dr. Hopkins, I know that's the UK is a very cost-conscious environment, and I know we've met with Professor Lund as well, and she's had an opportunity to trial the product in a couple of patients. I understand the challenges there, and what I would hope and what we hope is we find a way to get the product in all of your hands, and then you choose how it fits best into your own unique practices.
My question would be, you said it was a class type 3 medical device. Is that the same situation for you, or is that considered a drug device? Because that would be different to incorporate in our hospitals. If it's a device, we may have more problems. If it's something we can use like a packing at the end, which is drug, it would be completely different and perhaps easier to introduce, at least in our environment.
We are still awaiting FDA response,
Mm-hmm.
so I do not have detailed information on that
But
yet.
did you apply that as a drug, basically, or as a device?
Yeah, it's not something that I can really
You
go
cannot
into
disclose.
at this,
Don't worry.
at this point.
Don't worry.
Right.
I think the point here is that if it's a device, and yours is a device, not a drug, that's an advantage. It's much less studies, it's much less work in registration, so that's an advantage. And my first thought, well, great results, but honestly, did I expect something else? No, because you deliver topical corticosteroids, omeprazole, fluticasone, whatever. These results are more or less expected, to be very frank. What I would like to see is how does it compare to nasal douching or drops that we do otherwise. That's the key point. I think that's the study you should do. And you can even build in elements like comfort to the patients or something in that kind of study in order to see the differences. Some patients like douching. They even do, if I say no, they still continue to do, and some take whatever, honey or chocolate or whatever to their nose. But there are a lot who don't like that, and that's exactly the population there. The other thing which I found very interesting with your fibrinogen, I think that's not just a material to carry something, that has its own biological activity. And I would really like to know more about what does it do there. And I think, like I think Claire said it for the frontal sinus, draft 3 surgery, that is an indication for me where that would be possibly very, very helpful, because it covers immediately the bony part. Yep. And that could be extremely helpful if that's, you know, overgrown by epithelial cells immediately with the medication you release. And the other thing is, of course, as you said yourself, you know, there might be more than steroids. Doxycycline is one of these that helps to reduce metalloproteinases, and there are other candidates that have more functions actually in that situation. So I think there's much more music to still discover in these, and but what you need is really to show the comparison to what we currently have, not just versus placebo.
Certainly.
Wytske, would you like to comment something about the lateralization of the middle turbinate and the Propel device put in? It looked very nice on the long term, but we all know that lateralization of the middle turbinate occurs 3 to 4 months after surgery, not 30 days. Yeah. So would you see an indication to put a second device or even a third device to prevent lateralization there?
I'm personally not interested in lateralization. And when I really want to prevent lateralization, I put a stitch and stitch the middle turbinate to the septum, and then it won't lateralize. What I'm really interested in is getting high dose of steroids locally there, you know, because especially in the post-op period, because then you see a lot of polyp formation, probably just because we are there, probably because we make a trauma, that gives rise to extra, especially in severe patients, that's an issue. And then, of course, in the long period, because now a considerable number of percentage of our patients get systemic steroids a few times a year for a few weeks to prevent recurrence of polyps. And so at 2 moments, I would love to have better local treatment, not in the post-op period, but then long, preferably some months. Ideally, you would have something which would actually elute the drug for 6 weeks, 2 months, period like that. And then an easy product that you can move in between the polyps, either in operated or in not operated patients, or maybe in the olfactory cleft, where we all struggle with polyps, to have a high dose of steroids there and reduce the inflammation and polyp formation. So that are the needs I would like to have solved.
Informed correctly, this 30 days has a reason, and that's the FDA rules, saying if it's longer than 30 days, you have to follow other rules for registration. Is that true?
Actually, that is not true.
No?
For our initial device, we designed the excuse me, the 30-day period to align with the natural wound healing process. And we knew that with the mometasone furoate, if we delivered it over the course of 30 days, it's retained in the tissue. I think you saw that with fluticasone Madorna's data as well. We know those drugs have staying power, if you will. But what we wanted to do is provide mechanical support over the first 2 weeks to align with what was on the market at that time, which was spacing and spacers, which tend to function throughout approximately the first 2 weeks. And then continue to deliver drug throughout that initial wound healing phase, if you will. We chose not to go longer with our initial products because we also wanted to make sure we had something that was dissolving quickly and not staying around for such a long period of time postoperatively. That may or may not be the answer for every patient, but that was the rationale for our initial products. One of the products in development that I was speaking about that is designed to be placed within a polyp cavity in the office actually releases drug for 3 months and delivers over a longer period of time. But that was the rationale for the 30 days initially. And that second product, by the way, is being regulated by FDA as a drug. And for them, it's not necessarily the length of time of delivery. It's what they deem as the primary mode of action. And in the initial situation, they deemed the mechanical benefits to be the primary mode of action. In the secondary product, because it's for treatment of polyps, they deemed the medication to be the primary mode of action. And that was the basis for their decision.
Yeah. Of course, there's a difference in mode of action which makes a delivery or a drug, or it's a length. And I got that argument from the FDA once, and we developed a study over 3 months, and they said you have to change the insert every month because otherwise other rules apply.
Interesting.
And that was on doxycycline, by the way.
Sure.
So, yeah, Peter?
Could I raise a concern here from my side? In the post-operative care of patients after sinus surgery, we tend to advise to rinse and douche excessively the nasal and paranasal sinuses and cavities. What happens with the drugs that are eluted when we advise a patient to rinse at least 4 to 6 times a day with a massive amount of saline? Do we rinse the drug away, or do we speed up the resorption of the device? What is your opinion, or what are the data on that?
We have not seen an impact of nasal rinsing. The studies actually required it as part of the protocol. I think different physicians have different rinsing regimes, either 2 to 3 times or 4 to 6 times a day, whatever it may be. We encourage that broadly and haven't seen any negative impacts on the device. We actually believe that keeps the cavity more moist, which represents a better environment for that drug to continue to elute into the cavity. And then secondarily, again, one of the benefits of the drugs is that they are lipophilic, and they don't tend to want to get absorbed into water, rather into tissue. And that was one of the primary reasons for the efficacy of these types of drugs.
Does the question, does that issue concern your product as well? I mean, that is a little bit more stiff. It really expands and keeps in place. What about the thing you put on the raw surface? Would that be removed by rinsing?
When we started the clinical study for the first time, that's something we were nervous about. What is going to happen after? Is it going to withstand the wash? And it was okay, and it did not have any impact on the device. It remains in place pretty much organically. It becomes part of tissue pretty much immediately. We did not have that issue. We would not prevent physicians from doing what they would do normally. And to Susan's point, these drugs are so lipophilic, it's really hard to wash them out. So a rinse that would take, few seconds would probably not have a significant impact. Especially in our case, because the film gets epithelialized rather quickly, we don't think that would become an issue in the long run.
We have Metin.
First, we should separate the polyps. We should not apply this treatment to all the polyps. We need some polyps with eosinophilia. First this. And the second is a medialization process. If we want to medialize to prevent the lateralization, we put the stitches in the inferior part because we want the drug to reach the olfactory fossa, the upper part. Even if it is too medialized, we a little bit lateralize to give a space. And the second important point, these devices should not replace the surgeries. If you don't do good surgery, whatever you use, it does not help the patient. And these are not take the place of surgery because all these treatment modalities are after the surgery to the cavities. Yeah. Now here we have two questions to answer. One is cortisone, corticosteroids, prevents epithelialization. So we want to, the area should be epithelialized, but we put corticosteroids and we prevent epithelialization. So the answer to this is, I always say, we should preserve the mucosa as much as possible. So if we keep the mucosa and at least periosteum, it will re-epithelize, and corticosteroid help the edema in this respect very well. So I always say the diseased mucosa is better than regenerated mucosa. So the usefulness of these technologies, after opening the cells, it will give the surgeon the comfort not to remove the mucosa. So I can put this and it will help the edema to resolve. One is this. And the second thing, so the people will not take the mucosa. We should prepare to leave the bare bone. We should leave the mucosa. So the main idea of the surgery is to open the cells, but to keep the mucosa, leave it in place, and then these technologies will help a lot with irrigation, of course, together.
If you don't mind, so these are all excellent points. And actually, when we were doing our preclinical studies, we had all of these questions in mind. So we designed the studies where we completely would remove mucosa, would put the film on. Then we would compare it to an intact mucosa with the film on. And then totally remove mucosa just on its own and intact mucosa just on its own. And we compared them out to about 30 days, which was double the time that the film would actually reside. Mm-hmm. And we did not see any effect of our product in terms of either preventing wound healing or having any negative impact on the intact mucosa. So we really took that very seriously, especially with corticosteroid. Obviously, there are data out there that they could prevent wound healing, but the topics of the wound healing, they talk about the doses are in milligrams or sometimes tens of milligrams, where what we are bringing to the table in terms of local drug delivery, we are significantly reducing the dose, even compared to a douche, even compared to a nasal spray. We are significantly reducing the dose. So preventing wound healing, if anything, we would be able to help with that by reducing the dose, to your point, because then you use less of the drug because you're delivering it more efficiently.
Klaus?
I really don't have concerns about topical steroids. Also for long term, there's a study over 1 year showing very nicely that the epithelium doesn't suffer, just the opposite. It grows better and it grows to normal structure. So that's not a problem for me at all. With the other thing is, I think that for specific patients, you have to be more aggressive and you should leave the perios but take the mucosa away. And it's not my idea to leave that and then treat it. That would be the incomplete surgery, which is then saved or salvaged by putting these in. I don't think that's the good concept. I really see very nice possibilities to use it directly post-operatively, but also in cases where we see things get cobblestoned and comes back, and then you put it in there, and then you could have a better delivery. And the question really is, and also with your Propel, how much steroids do you find there if you combine it to if you compare it to a topical steroid spray or a solution or drops? And what is that? Is that the same level? Is that higher? Is that much lower?
I think the question is where you're looking. So if you're looking within nasal cavity, probably the amount of steroids from nasal spray is much higher, because for most part it deposits in the nasal cavity and not a whole lot of it actually gets to the sinuses.
Locally in the mucosa where you actually apply it.
So in our case, we are actually applying it right in the sinus cavity. So that's kind of where some of our animal data really clearly show that, where we look at the mucosa inside the sinus, we'll look at the nasal cavity, and I think this has been your experiences as well. Much higher doses within the sinus and much lower doses within the nasal cavity. Basically, that's what we are trying to do, to get the drug to where it really needs to be, rather than having a lot of it swallowed or deposited places that you necessarily don't need.
What about systemic uptake? If you compare that to I think when FP came on the market 15 years ago or so, we didn't have these really sophisticated ways to measure. So then I remember we tried really high doses up to 12 milligram of FP in a spray form and couldn't even find it back in the systemic circulation. But do you know, are those data now available comparing, say, drops or spray to your device?
Obviously, they haven't compared to our devices specifically, but we can look at the data that is available. So FP is very well studied, so the data is available, and usually is very, very low systemic exposure. And the levels that we are delivering, really there is no way there would be any systemic exposure to the patients. And I think to your point, really at the end of the day, our goal is to provide a technology that when you do a perfect surgery, then you don't go home and worry about the cobblestone coming back. So you know that you did a safe addition to what you just did, which was a great surgery. The patients would not have any side effect from it. If anything, their outcome would be improved. You have a peace of mind that they actually they received the care that they needed to prevent them from those inflammation coming back. And this is some of the areas that I think your data and our data kind of follow each other, that especially in subgroup of patients with nasal polyps come back aggressively and quickly. They come back really quickly. A lot of other literature, they look at 6 months, 3 months down the road, which is really relevant. But there are not many literature that look at early return of polyps, and they're significant. There is a significant percentage of return.
What you directly have after the surgery is the post-operative phase, where a lot of edema is not polyps. So we are talking about really, as you said, what is about after 1 year? Does that still have an effect? I would probably guess no, but I would see the chance to reintroduce it if it's needed. I mean, that's exactly it.
How do you see in the future a study that would convince us to start using it? Like a study where you would compare a regular nasal steroid spray with your devices. I think that would be the most convincing study for me to start engaging on using it or considering it for using in clinical practice. Do you have this type of study in your portfolio or pipeline, or is this not something you can disclose?
I can share what we have, and we have included nasal sprays in the control arm of our studies to ensure that that represented the typical postoperative care. In our initial studies, they were not allowed for the first 14 days, and that was done because in our research, and we talked to clinicians, they tended to not prescribe them in, starting day one after surgery. They were waiting for a period of time. In addition, the nasal sprays weren't indicated for that immediate postoperative period. But we did allow for nasal sprays in the control group, and so the differences that you see, especially in our frontal study, represent that.
What I really like from a surgery perspective is that we do not rely on the compliance of the patient of nasal spray. We put in a device that is drug-eluting, and actually compliance, which is lacking in 50% of the patient, is actually not an issue anymore. So that holds a major potential for us.
That's
I
a
think.
good point. Yeah, Metin?
I think for the future of these technologies, one of the most problematic area is maxillary sinus, because in diffuse nasal polyposis, the maxillary sinuses, the topical drugs, do not reach these maxillary sinus. If we can use this for the maxillary sinus, it will help a lot, because it's a lot of polyps and polypoid and edematous areas. We create mega ostia for the drug to reach the maxillary sinus. So if you use this device also for the maxillary sinus, it will help a lot. And the second point is for the studies in the future. If you look at the studies, they look at the lateralization, polyp, etc. They are not very clear studies. We should organize studies, one side the technology, I mean corticosteroids device, and the other side placebo. So if you look at the literature, only there is one such study, and it's also should be more sophisticated.
I think I would like to turn the table to you guys now, is to help us, like similar discussions you had with previous panels, to support in terms of clinical study designs. Obviously, a lot of studies have to be done separately because of confounding factors and kind of the evolution of a technology, but definitely having more guidance and support from academia and support in terms of kind of identifying, and what you have done with the European position paper of what are the guidelines and what we should take as this is the standard of care and what do we really compare against. I think these are all going to be extremely helpful to us as we design the studies to bring to you guys and show you the outcomes.
I would really like to see, apart from spray, where Metin already says it really hardly reaches anything. It's a miracle that spray actually work. I think more and more of us use rinsing with saline with steroids in there, with drops or budesonide or FB. So I would really be interesting to compare it to that, because I really feel that that is more effective, and I think there's now also a study showing that. Because if you there's quite a lot of literature actually showing that if you get local steroids in the sinus, it's much more effective than when you spray it in the nose. So, and of course, if the device is as good as rinsing, it's probably still worthwhile, because you don't need compliance, and not all patients like rinsing. But so that will be a very interesting comparison, not only to spray, but actually to rinsing with saline and steroids. Although I understand the difficulty that it's not approved in that way.
Yeah, so, which has been the challenge.
Yeah, but still, from a clinical point of view, that would be really relevant.
And I would also, if I may add, look at a little bit of different concepts according to the endotypes we have now. So you're not dealing a chronic rhinosinusitis or treating a chronic rhinosinusitis without nasal polyps the same as polyps, and then within the polyps there are different groups. Mm-hmm. We discussed this morning. So I think there are different possibilities also to use either post-operative, independent of eosinophilia, or in those patients with recurrent disease, and then you are looking to a totally different inflammation, and you don't look at a totally different patient population, and there might be different indications. So I think you should differentiate your studies in the future a little bit more according to these different types of diseases.
So I'd like to thank both of you for your nice presentations, and I'd like to thank the panelists for their contributions. I think we have seen two very promising products which need to undergo yet some future studies, but the products look really promising, and I'm looking forward to see its application in the sinuses or even elsewhere, which we have discussed previously. Thank you very much.